All right, class. As I mentioned, this is a big chapter and very important, so please play with me. I apologize in advance. This is a long chapter, but it's really fun. There are more things to learn from this chapter. So next we have digestion, absorption, and transportation of carbs. So, this is the anatomy of our digestive tract or digestive system. Our digestive system starts from mouth and it ends in anus. And different organs and tissues are all involved in the digestion and absorption and transportation of carbohydrates. We have salivary glands, esophagus, which is between the mouth, like between pharynx and stomach. We have stomach, we have small intestine, large intestine, we have liver, gallbladder, pancreas, all these different organs. We have bloodstream, we have lymph system. All these organs and tissues and different systems are involved in digestion and absorption and transportation of carbohydrates. So this is a picture of our small intestine. As a sport nutritionist or as a sport dietitian, I would recommend to know like some basic anatomy of the digestive system. So it's very important. A small intestine has three sections. The first section is called duodenum. The second section is called jejunum, and the last section is called ileum. And then we have large intestine. Again, large intestine has three or four different sections. We have, um, like from here, we have ascending colon, transverse colon, and descending colon. We have sigmoid and rectum. Those are different parts of a large intestine. So it is really important to know the anatomy of digestive system. And the small intestine, it's highly it's highly like branched, it's kind of like very tiny hair-like structures. The surface area of a small intestine is equal is like is equal to a two-car garage. So it's a very large surface area. In order to optimize the absorption rate. Um, in this picture on the right side, we have villi. So these are like these structures, like hair-like structures that increases the surface area of a small intestine, and the absorption happens in these epithelial cells lining the villus. So villi is the plural of villus, and we have like cells in one villus, different cells in one villus. So here is the digestion of carbohydrates. Actually, this picture, the top picture shows like the digestion, and the below picture, the bottom picture shows the absorption. So we have digestion and absorption in this picture. So, when we eat food and the food is, let's say, high carb meal or a meal that contains carbohydrate, which can be fruits, vegetables, dairy products, the digestion of carbs starts in the mouth. We have salivary amylase. Amylase is an enzyme in our saliva that starts breaking down carbohydrates. This enzyme gets inactivated in the stomach, so we don't have any digestion of carbohydrates in the stomach because of the acidic environment inside the stomach. But in the small intestine, we have pancreatic amylase. So this is the enzyme that is produced and secreted by the pancreas. We also have enzymes, different types of enzymes in the brush border of epithelial cells. So we have like the lumen, and we have brush border uh, of these small intestine cells. So this brush border also produce some types of enzymes, so lactase, maltase, and sucrase, and the sucrase also digest maltose. And also we have some conditions called lactose intolerance, which we will discuss at the end of this chapter. So remember this. So digestion of carbs begins in in the mouth because of the salivary amylase. There is no digestion of carbs in the stomach, and in the small intestine we have pancreatic amylase, and different enzymes are produced from the brush border of the epithelial cells of the small intestine. So what we have in the small intestine after digestion of carbs, we have fructose, glucose, galactose, and some small chain uh, carbohydrates like disaccharides. So this is what we have. After this, we have to absorb these small one molecule of carbohydrates or monosaccharides. So the thing you need to remember and learn from this picture, fructose uses facilitated diffusion, and it uses a carrier called GLUT5. So fructose is absorbed through facilitated diffusion. Glucose and galactose, they use a secondary active transport system to get absorbed. And the difference between active transport and the facilitated diffusion is this. In the active transport, we need energy. So we need energy to absorb glucose and galactose. But regarding the fructose, we don't need any energy to absorb fructose. After they get into the inside of the cell, so we have fructose, glucose, and galactose inside of the cell of a small intestine. They can get absorbed to the bloodstream. So they are moved to the bloodstream through another carrier called GLUT2. And this GLUT2 is a facilitated diffusion. So we don't need energy to move glucose, galactose, and fructose from small intestine to the bloodstream. And from the bloodstream, those absorbed monosaccharides, they go to the liver. So this is the difference between absorption of fructose from other monosaccharides. The majority of digestion takes place in the small intestine. The majority of absorption takes place in the small intestine. So the small intestine is the major place for both digestion and absorption of carbohydrates. We have metabolism of glucose. When we say metabolism of glucose, it involves different pathways. It involves the regulation of blood glucose concentration, the immediate use of glucose for energy, the storage of glucose as glycogen. These are all part of the metabolism of glucose. We also use glucose for fatty acid synthesis. Fatty acid is a type of a lipid. We also produce glucose from lactate amino acids or glycerol. So these are all part of the metabolism of glucose. And when we say metabolism, it means all of the physical and chemical changes and reactions taking place within the cells. That is referred to as metabolism. For the metabolism of glucose, there are two main hormones. There are other hormones that can affect the metabolism of glucose. For instance, we have epinephrine, norepinephrine, but insulin and glucagon. These are two main hormones that regulate the blood levels of glucose. These two hormones, insulin and glucagon, are produced from pancreas. The normal blood glucose level should be between 70 to 100 milligram per deciliter of blood. We have hyperglycemia. Emia means blood, hyper means high, and gly means glucose. So when we say hyperglycemia, it means high blood glucose levels. We have hypoglycemia. Again, emia means blood, gly means um, glucose, and hypo means low. So it means hypoglycemia means lower or low blood glucose levels. And we get hypoglycemia whenever the blood glucose levels drops to 50 milligram per deciliter. And we have homeostasis. Homeostasis is a term referred to as an state of equilibrium. So the insulin and glucagon tries to maintain the homeostasis of our body. So this figure shows the function of insulin and glucagon. It depends on the amount and type of the carbohydrates. The rise and return of blood glucose may take 30 minutes to 1 to 2 hours. So if we get a simple carb, the rise and return of blood glucose happens very quickly. But if we get complex carbohydrates, it may take 1 to 2 hours. So here is what happens. We have whenever we eat carbohydrates, that carbohydrates enters to the bloodstream as glucose. And that glucose stimulates beta cells in pancreas. Pancreas releases insulin to the bloodstream and it forces other cells to take up the glucose from bloodstream. So it drops the blood glucose levels. In fasting or starvation, we don't have much glucose as we have after eating. So that fasting stimulates alpha cells in the pancreas. And those cells, alpha cells, produce and releases glucagon to the bloodstream. And glucagon makes the liver to release its stored glucose as glycogen to the bloodstream. So this is all about the function of insulin and glucagon. We have GLUT4. These are for glucose, there are different carriers and vehicles, I would say. These are the carriers that glucose use to travel from outside of the cell to the inside of the cell. So we have GLUT4. We see GLUT4 in skeletal muscle and adipose tissue. And GLUT4 is insulin dependent, which means without having insulin in the blood, GLUT4 is not working. So we need insulin to activate GLUT4, and glucose uses GLUT4 to transport. Brain cells and liver cells and exercising muscle, they are not dependent on insulin. So at rest, skeletal muscle depends on insulin to take up glucose from bloodstream. But during the exercise, the muscles do not depend on insulin to take up glucose from bloodstream, and the brain cells and liver cells. We have different carriers again for glucose on brain cells and liver cells, but they are not dependent on insulin. We already talked about like the glycolysis and oxidative um, system in previous chapters, so I'm not going to talk about that here. But remember, like glucose or glycogen is broken down to pyruvate, and the pyruvate can be converted to lactate or oxidized. So we have anaerobic metabolism and aerobic metabolism. And regarding the glycogen, skeletal muscle contains 400 gram and liver 90 gram of glycogen. So muscle cells have more glycogen than liver. But there is a slight difference here, and this is very important. The glycogen in skeletal muscle, they are not released to the bloodstream. So whenever skeletal muscle breaks down glycogen, it uses the glucose for itself. But liver breaks down glycogen and releases glucose to the bloodstream. And the excess glucose in liver and adipocytes, they can be converted to lipids. All of these foods on the picture, we have bread, rice, pasta, legumes, contain carbohydrates, but they each have a different effect on blood glucose. Why is this? How might this affect an athlete's carbohydrates choices? So here's a question for you. We said different foods like grains, legumes, vegetables, starchy vegetables, non-starchy vegetables, fruits, they all contain carbohydrates. But the important thing is these carbohydrates have different effect on blood glucose. Why is this? So here is our answer. We have glycemic response. This is the term referred to the effect that carbohydrates foods have on blood glucose concentration and insulin secretion. We have glycemic index. I know you remember from a table at the beginning of this chapter. There was a table on monosaccharides, and we had glycemic index for different monosaccharides. So, and I said I will discuss and I will explain it here later. So here it is. We have glycemic index, which is a method of categorizing carbohydrate containing foods based on the body's blood glucose response after their ingestion, digestion, absorption. Glucose or white bread, which is a refined bread, containing 50 gram of carb is used as the reference food in this method. So here is the formula for GI. We have our AUC test food and AUC reference food. AUC is the area under the curve of test food divided by the area under the curve of reference food multiplied by 100. This gives us GI or glycemic index. This is how they measure glycemic index. So here, this is an example. We have applesauce. Applesauce is a low glycemic index food, and we have orange juice. Orange juice is a moderate glycemic index food. So the difference between these are shown in this figure, in this graph. So after eating applesauce, the blood glucose levels increases, but it's lower compared to that increase induced by orange juice. So we can conclude based on this graph that applesauce has is a low glycemic index food, and orange juice is a moderate glycemic index food. We can also categorize different foods based on their glycemic index. We have high glycemic index foods whenever the GI is more than 85. For instance, white bread, baked potatoes, starchy vegetables, and cornflakes. And we have low glycemic index foods, which is GI less than 60, including legumes, beans, fruits, and not non-starchy vegetables. Gluconeogenesis is also part of the metabolism of glucose. Gluconeogenesis is a term meaning and creating and making a new glucose. So gluconeogenesis is the process of producing glucose from other sources. And these are the different sources that liver, most of the time, the liver uses different substances, including lactate, amino acids, glycerol, to produce glucose. Lactate and amino acids are the major sources to produce glucose. So we have use of muscle glycogen during exercise. As I said, liver use glycogen. It breaks down glycogen and releases glucose to the bloodstream when we are fasting when we are starving, when we are exercising. In our post-meal state, liver breaks down glycogen and releases glucose to the bloodstream. But muscle cells breaks down glycogen during exercise and they use the glucose from glycogen for their purposes, for their energy. And this is what happens for the glycogen content of a muscle tissue after exercise or during exercise. So, for instance, here in this graph, the muscle glycogen at time zero, before starting the physical activity or exercising, it's close to 1.5 gram per 100 gram of the tissue, muscle tissue. That's the concentration of glycogen in the muscle tissue. And during exercise, we see a drop in the levels of muscle glycogen. And after like 60 minutes or close to 1 hour, the concentration of muscle glycogen is close to almost zero. And this is where the fatigue happens. So exercising muscle prefers glycogen over glucose. The exercising muscle cells, they prefer to break down glycogen over just using simple glucose for their energy source. In moderate to high intensity aerobic exercises, carbohydrates and glycogen is the primary energy source. Or in higher intensity and repeated intervals, our body, our muscle use glycogen over fats and other energy sources. And the rate of glycogen usage depends on exercise intensity. So in higher intensity exercise, the rate of glycogen usage is high. And the fatigue happens when muscle glycogen depletion happens during prolonged endurance exercise or during intermittent high intensity exercise. Which statement is incorrect? So this question asks about incorrect statement. A. Exercise has a strong insulin-like effect, meaning exercise stimulates glucose uptake from the blood. That's true. That's correct. Exercise has an insulin-like effect. Glycogenolysis, which means breaking down of glycogen, is the release of glucose from glycogen caused by glucagon. That's also correct. Glucagon is a hormone produced by pancreas, causes glycogenolysis, specifically in liver. Glucose is preferred over glycogen. That's incorrect. Glycogen is preferred over glucose. And the last statement, muscle glycogen utilization is no different when athletes consume a carbohydrate drink. compared to a placebo drink during high intensity activity. That's correct. So blood glucose during exercise. As I said, exercise has an insulin-like effect. Exercise stimulates the uptake of glucose from the blood by skeletal muscle. So it decreases blood glucose levels. Dropping blood glucose levels stimulates pancreas to produce glucagon. And glucagon stimulates or induces glycogenolysis, which is the breaking down of glycogen, or it can induce gluconeogenesis, which is making new glucose from lactate or amino acid or glycerol. Regular exercise training. So when we exercise regularly, when we train our muscles regularly, specifically aerobic training, which is using oxidative system or aerobic system, we are increasing oxidative enzyme activity. We also increase number and size of the mitochondria of muscle cells. This can lead to an increase in oxidative capacity. And this increase in oxidative capacity can lead to increase in fat metabolism for energy or muscle's total capacity to utilize carbohydrates. And also, when we regularly exercise or when we train regularly, we also increase the stored muscle glycogen levels or concentration. And from a higher stored muscle glycogen, we can increase the muscle's total capacity to utilize carbohydrates. So resting, this is really interesting, right? Resting muscle glycogen in average sedentary adults are 20 to 30% lower than those of trained athletes. This is really interesting. And 20 to 30% is a huge number. So this shows how exercise and engaging in different physical activity can train our muscles to store more glycogen and to use more oxidative system or aerobic system to utilize more fat and more carbs. So for regarding the glucose metabolism, as I said, there are many different hormones. Hormones can regulate the metabolism of glucose during exercise. So the glucagon uh, is secreted during periods of fasting, starvation, and exercise. So exercise is also like fasting and starvation. And exercise intensity affects carbohydrate metabolism. As exercise intensity increases, the proportion of energy derived from fat decreases and from carbs increases. This is really important to remember, a very important point. And the carbohydrate is the main source of energy for moderate to high intensity exercise. This graph is from your textbook, and it is derived from one study. They compared the placebo and the treatment between two groups. So for one group, they just gave them placebo, and for another group, they gave carbohydrates, and they measured the muscle glycogen levels. So it's as you can see in the treatment group, the muscle glycogen levels were higher than the control group, which were who were given placebo, and they start decreasing because of exercise. But the important thing to note here is to see whether the difference between treatment and placebo group was significant or not, which we cannot conclude just from seeing and eyeballing this graph. So remember when we um, try to explain or try to interpret the results of a study. We want to see whether there is a significant effect or difference between groups. But here we can conclude that when we are given carbohydrates, it increases the muscle glycogen levels. That's all we can conclude. And for the carbohydrate or treatment group, the muscle glycogen levels decreases just like the placebo or control group, but it yeah, like here in the placebo group in time 3, the muscle glycogen levels decreases stops, but for the control group it continues. So this is a really interesting study, I guess.